whatever propensities are present, both sexually and behaviorally, depends upon the presence of estrogen receptors. And once again, when those estrogen receptors are not apparent or not appearing in the male brain at the appropriate developmental time, the male proclivities, the male propensities do not develop normally. So it's curious that, again, it's the estrogen receptor that is more important for the male brain differentiation than T or DHT receptors. So we were talking about descent of the testes into the scrotum. We've indicated normally this can occur anywhere from a couple months after birth to a few weeks. A couple months before birth to a few weeks after birth. And they begin up here, as you can see, in the abdominal cavity. In fact, this entire reproductive physiology of both males and females begins this process of elongation and descent. That accounts for that crazy elongation of the vas deferens over the back of the urinary bladder, for example. I mean, that's just a crazy engineering design there. But in any case, this has to happen. This descent has to happen. It's better that it occurs before birth. If it does not occur in a timely fashion, it does not occur shortly after birth, that is almost always associated with increased risk of various pathologies, including, for example, testicular cancer. There are various methods by which this can be done by palpation. The pediatrician can sometimes palpate it, and all sorts of surgical procedures to affect that descent. That does not reduce the risk of those diseases, those pathologies. So even though the testes can be brought into the scrotum artificially, the disease risk remains fairly high in those individuals. All right, we're looking at this what's called the spermatic cord, an incipient spermatic cord here. This would be the structure that, or from which the testes are suspended from the abdominal cavity. And we'll talk about the contents of that spermatic cord in just a moment. But there are a couple of muscles you should be familiar with that contribute to the regulation of the scrotal temperature. Remember, in the animation, he pointed out that spermatogenesis, development growth of the sperm, depend upon the temperature being a little bit lower than the body temperature. Well, in that spermatic cord up here, we have what's called the cremaster muscle or the cremasteric muscle, and that is a sort of a wispy, discontinuous cylinder of muscle about the perimeter of the cord that is mostly important for elevating the scrotum with respect, elevating and moving the scrotal sac relative to the body. That's the cremaster muscle, which, as I say, is more of a cylindrical tube in the spermatic cord. And then the scrotal sac also has a layer of muscle called dartos muscle. And this is the object of so many late night jokes. This is the one that regulates the skin tension around the testes. And so the two together have to maintain the appropriate temperature as close as possible. Of course, clothing would play a role in this, perhaps behavior, but to the extent to which you ride a bicycle, that sort of thing would all contribute. But in any case, you see the dartos muscle would be out here, cremaster muscle would be in the suspensory tissue. If you've ever watched a cow or a horse, when they get flies landing on their back, what do? In their skin. They have the tail. But they can also, if you notice, they could shiver their skin. And so we could argue about this, but the dartos muscle is about the only analogous tissue, the only analogous such muscle in humans, where there's actual attachment, smooth muscle attachment to the skin, causing that type of change in the tonus of the skin. We'll talk about the peritoneum later. I don't know that I want to get into that discussion, but arguably the peritoneum is a complex membrane system that lines the abdominal cavity. And you can see that what's happening as the testes sort of descend is they're pulling portions of the peritoneum with them, so that in fact you find that in most discussions, portions of the peritoneum surround the testes. So let's look at this anatomy that he mentioned in that animation a little more closely. We can see the testes here, sort of, and within each of these testes, there are lobules containing densely coiled seminiferous tubules. And these are the seminiferous tubules here, and of course they derive their name from the fact that this is where the semen, more importantly, the spermatocytes undergo spermatogenesis, originate and undergo differentiation. There are about 250 of these lobules. You only see a few here, but there are about 250 of these lobules, all each separated by the membranes or septa that you see in the illustration. The seminiferous tubules by far comprise most of the mass of the testes, and we'll talk about some of the other associated cells in a moment. Before we do that, let's make sure we understand the basic anatomy of this system. So here we can see the coiled seminiferous tubules. And then, as they emerge from the lobule, they give rise to these structures right here called straight tubules. And the straight tubules lead to this structure, which is called the ret. Ret just means a net-like structure. There are various rets of the body. A lot of them are vascular. And then, as you can see, emerging from the ret are these structures called the efferent ductules. I'm not very good. There's a little displacement between the pin and the... But in any case, those are the efferent ductules right there. And of course, the efferent ductules are then giving rise to the epididymis. That would be the head of the epididymis up here, also coiled tube. This is the body. This is the tail of the epididymis down here. And then that epididymis is going to reflect or fold back to form the ductus deferens or the vas deferens. So you'll see both terms, ductus deferens or vas deferens. And then that's that crazy structure that we saw go back into the abdominal cavity, then arch up over the urinary bladder. Now we want to talk a little bit about the histology. This gets a little bit more complicated than we need to get into. But again, before we do that, you can see in this illustration the seminiferous tubules sort of packed into here. You can see a bit of the ret and the efferent ductules, the ret here, and the efferent ductules up here, and then here's our epididymis giving rise to the vas deferens. And as far as I know, there's no precise transition point. It just sort of becomes the vas deferens when the degree of coiling tends to diminish. And as we're going to learn, we learned about the stages of spermatogenesis. For example, we talked about spermatogonia, primary spermatocytes, and second. All that's happening right in here. So we want to talk a little bit more about that. And that's what this illustration over here is introducing for us. This is sort of a slice. This is a part of the wall of the seminiferous tubule. So you can imagine the seminiferous tubule here. There's the lumen in the center. This is just a chunk of it right there. And this would be the lumen out here. And that would be the wall. So just maybe turn it upside down. I guess I should have drawn it down here. So out here at the perimeter of the seminiferous tube, you'll notice what you have. You have the stem cell that gives rise to these suckers. This is the spermatogonia. And then there will be progressive differentiation of the spermatogonium into the spermatid as that cell, once it's differentiated, migrates toward the lumen from the perimeter of the seminiferous tubule. So these cells are just going to be moving in this direction, and as they do so, they undergo progressive spermatogenic differentiation. So again, just sort of to go through some of these terms before we get into the details. Several things have to happen in order for this process to be completed. There must be an adequate supply of spermatogonia. And if you recall, we said that the stem cell has to do one of two things, actually has to do both, but at any given point in time, it's doing one or the other. It has to make more of itself through mitosis, or it has to begin the process of differentiation. And in the process of differentiation, that spermatogonium now becomes a primary spermatocyte. Notice there's no division in this transition. There's no division in going from here to here. It's just begun the process of differentiation. It's differentiating into the specialized cell. All right, and so then we're going to review the spermatogenesis sequence again. We've already learned that, but we'll go through it again. And then you'll notice at the here at the lumen, at the margin of the lumen, you have these oblong, odd-shaped cells. These cells are undergoing the process. The technical term is spermiation. I'm not sure that this is quite the correct definition here. But spermiation just means that these oblong cells, they kind of look like a heart shape there, that they become spermatids from this round cell. And then the final process, so let's just sort of list them here so we don't get confused with the terminology. There's spermatogenesis. Then there's spermiation. And then finally, that's probably not correct, had provided. There is spermiogenesis. So spermatogenesis just means we're going from spermatogonia to spermatids. So that means meiosis. Spermiation is the process whereby the spermatid acquires its more or less normal functional shape. We'll see what we mean by that in just a moment. And then spermiogenesis, there's a couple definitions, but generally what we mean by spermiogenesis, and this takes much longer, it can take up a couple months or so, is the process of the spermatid acquiring motility. And what do we call a spermatid with motility? Anybody know? Spermatozoa. Zoa implying the motility. All right, any questions about the general definition of those terms?
What about spermation, Dr. King?
Spermiation is this process right down here. So by spermiation, we generally, and again, I don't really like what I've written here, so maybe that's not the best way to express it. Spermiation implies that see how technically a spermatid, technically a spermatid is round. And you can see as this round cell, see they have it labeled here, spermatids, but as they acquire their shape, and these pink cells, we need to talk about these pink cells. But what these pink cells do, part of their job, they have a variety of jobs, but part of their jobs is to gobble up all of this extraneous cytoplasm and turn this cell into this cell into this cell. And the process where we go from this to this is spermiation. It's just the spermatid acquiring its what's the word I'm looking? Its functional shape. It's not yet functional, but it's acquiring the functional shape. So here's a transection of a seminiferous tubule. So make sure you understand we're looking at the seminiferous tubule here. So here's the perimeter out here for us, and that's where you would find the spermatogonia at the perimeter. And this is one of these famous Dennis Kunkel. I don't know if he's still alive, but he was famous back in the day because he enforced his copyright, and if he ever found anybody with his images, he'd go after them legally. So you're not supposed to tell him. I don't know if he's still alive. These fairly old images. But this is a colorized representation. Actually, it's the real thing, but he's colorized. And here we can see the spermatocytes undergoing differentiation. And then in the lumen, we begin to see the more or less appropriate shapes of the spermatozoa. We have to be careful here because, again, spermatozoa implies motility. In the seminiferous tubule, these cells do not have motility. They just look like they do. They have the flagella. We saw that animation, the flagella was spinning like a propeller. But at this point, in the seminiferous tubules, although they look like spaghetti with flagella there, they're actually moved along the tubule by peristaltic contraction. They don't swim on their own at this point. And of course, over here on the right, we have an idealized representation. So I just want to make sure you understand the general sequence of events. So out of the perimeter here, we can imagine this as being a spermatogonium. It's going to undergo differentiation. You can see some cell division, some mitosis going on there. It's going to become a primary spermatocyte, then divide to become a secondary spermatocyte. That'll be the first division. And then what happens to the secondary spermatocyte in the second division of meiosis? What does it become? So we start out with the spermatogonium. It differentiates into a primary spermatocyte. The primary spermatocyte divides to become secondary spermatocyte. So that's meiosis 1. And then what happens in meiosis 2? The secondary spermatocyte becomes a spermatid. And all of that's happening right in here as it's moving from the perimeter to the lumen. And then it looks like yachts docked at the marina there. You can see the spermatids lined up at the lumen. Now, we haven't talked about this very, very important cell back here. And in this illustration back here, it's these pink cells. These are actually large, massive cells. They go by a variety of names. And in this image, it's this, what is it, purple here? So that's a cell there too. So all of that you see is purple. That's one, well, let's be careful here. Let's see if we can find a cell junction. I really don't see it in this illustration. You can sort of see one right there. If you squint, can you see that line right there and that line right there? So we have a cell here and a cell here, and they're separated by that line. But what I want you to understand is this whole thing then is one cell. This whole thing is one cell. So how would you describe, you can tell I'm struggling with my words today, how would you describe the relationship between that large pink cell and the blue cells? I'm sorry? I think so. I think that's what I want you to get out of this. The blue cells, the spermatocytes, are not inside the cytoplasm of these cells. These are going to be called Sertoli cells. But as Bob suggests, the Sertoli cells envelop them completely. It may not be one Sertoli cells, but collectively, all of the Sertoli cells surround and envelop the spermatocytes as they undergo meiosis. And that's absolutely critical because these cells, these Sertoli cells that you're looking at here, those purple cells, are the mother cells, if you will. They are the nurturing cells. They're sometimes called the sustentacular cells because they sustain the developing spermatocytes. And they do that by creating the proper chemical environment for meiosis to occur. So the whole job of those large Sertoli cells that you see here it is again right there. All that light-colored area that you see out here, those are Sertoli cells, and the dark areas would be the spermatocytes, or at least the nuclei of the spermatocytes. So their whole function is to create the proper environment for development and differentiation of these spermatocytes. Although you can see a cell junction there too. And also to create a protected area that prevents immunocytes, immunoglobulins in the circulating blood from getting to these cells. Sertoli cells are said to be immunologically privileged. Trent, did you have a question?
Yeah, so the dark purple is the nucleus of
Yeah. Any questions then about the general physical relationship between these Sertoli cells? Very, very critical. We're going to talk a little bit more about the specific functions, but I want to make sure now you see the relationship and see now again in the actual micrograph over here how these cells are, how these spermatocytes are moving in this direction, progressively differentiating, but all, as that happens, very much dependent upon the proper environment being maintained by the Sertoli cells. These cells are special, as I say, they're immunologically privileged. I worked with a fellow at USF many thousands of years ago, and his big idea was to take these and use them sort of as liposomes. He would take these, he would put, for example, neurons, package neurons into these things and put them in the brain of patients with Alzheimer's or Parkinson's disease. Why did he do that? He would take neurons from one patient, not somebody living obviously, but take neurons from one patient and put them into the brains or even another location, into the brain of a region, let's say the substantia nigra, some other region that might be compromised. Under what theory, under what premise? These cells would prevent rejection. These cells would prevent any sort of, it didn't work, but that was the theory. And they've been used in a variety of similar ways in more recently. So this is just a review of the meiosis that we've talked about. Hopefully you understand the sequence of events now. We talked about the spermatogonia, which is going to be out here at the margin of the tubule. Be sure you understand, folks sometimes have trouble with this. In order for this spermatogenesis to occur, in order for it to keep going, did you happen to notice in the animation how many of these spermatocytes are being cranked out each day? Yeah, I mean, just millions and millions. So in order for that process to continue as it does in that insanely hectic rate, these spermatogonia must be able to replicate themselves through mitosis, and they must also, that pool of spermatogonia must also begin the process of differentiation. So really both processes are going on simultaneously. Let's not get into the details of which process is occurring first and which cell is doing which. I'm not even sure that's been entirely sorted out, but both processes have to be concurrent for this rate of sperm production to be sustained. So that's all this is, and then we've already talked about the point at which spermatogenesis begins in males, and when it concludes. When does spermatogenesis conclude in males? Yeah, it just goes right on to the end. We saw the fellow, 96 years old. All right, so we talked about the sequence again. Again, spermatogenesis just refers to meiosis. Meiosis 1 is the division of the primary spermatocytes into two, and this is very important, haploid secondary spermatocytes. And when that happens, the homologs separate. The maternal portion separates from the paternal portion. And then in meiosis 2, the haploid secondary spermatocytes separated into two spermatids. And at that point, chromatids separate. Notice, as we pointed out before, in male gametogenesis, 4 gametes are generated. In male gametogenesis, 4 gametes are generated. In female gametogenesis, only 1 gamete is generated. And once again, you see the micrograph here, the spermatogonia out here, progressive spermatogenesis, meiosis to form the spermatids. So we've talked about spermatogenesis, we've talked about spermiation, and let's look a little bit at spermiogenesis. The important thing to keep in mind, this is a long process. It can take a couple months or more to be completed, and this process is completed. The process of the spermatid acquiring motility and functionality takes quite a long period of time and occurs as the spermatid leaves the seminiferous tubule, passes into the epididymis, and then moves along vas deferens. As you can see, it spends about 12 days in the epididymis. It says here to full motility. Now let's make sure we understand what that means. It means it, in theory, has the ability to propel itself. It really, the spermatozoa really do not become motile until this process is complete. And even then, the spermatozoa really doesn't start to, let's say, doesn't turn the engine on until it enters the female reproductive tract. And then we talked about the large number of these that are cranked out on a daily basis. So as you can see, the product of spermiogenesis is something like what we see over here on the right. There's an head with a very, very tightly, very, very intensely coiled DNA. At the apex of this warhead here, you can see a region called the acrosome that is chock full of hydrolytic enzymes. And those hydrolytic enzymes afford the necessary corrosive activity to pass through the egg cell. The region down here called the midpiece is chock full. This is sort of the engine room here. This is chock full of mitochondria and short supply of fuel to ensure that the flagellum, which is the final component, is capable of spinning and motility. And the motion that you saw in the animation is actually what happens. It's more like a propeller. It's just basically turning. And if you want to go online and look at the molecular biology of this little engine, it's incredible. It looks like a series of cogs and wheels all meshing together to create that motility at the molecular level. But in any case, the acrosome, the head, the midpiece, and the tail or flagellum form the spermatozoa. This has a fairly short lifespan, typically only 2 or 3 days in the female reproductive tract. Of course, for that matter, the egg doesn't survive very long either. So it's just a very limited window. Here you can see the successive stages from the spermatid, the round spermatid that we talked about. You can see spermiation beginning in the top left there. And then when you have this, again, theoretically motile creature on the right, spermiogenesis would be completed. Spermatozoa, or sperm, I guess, would be the appropriate abbreviation as well. So we're looking at a scanning electron micrograph of some spermatozoa. I'll be honest with you, they don't look all that healthy. And of course, when assessments are done of the health of the spermatozoa, they don't necessarily pay attention exclusively to the count. They also pay attention to the shape, the morphology of the spermatozoa. So an individual could have a normal sperm count, but could have malformed or dysfunctional spermatozoa that might contribute to their impaired fertility.
Spermatogonia are the cells from which sperm cells arise. The spermatogonia divide by mitosis. One daughter cell remains a spermatogonium, and the other becomes a primary spermatocyte. The primary spermatocyte divides by meiosis to form secondary spermatocytes. Secondary spermatocytes divide again to form spermatids. The spermatids differentiate into sperm cells.
And they're off. Now, you may want to be thinking about the numbers, and what those numbers refer to, and why were they designated in that fashion. So we talked about the spermatic cord. We're looking at it here. We're looking in the anatomy class. As time permits, we'll talk about the inguinal canal, and on either side, the spermatic cord will emerge from and descend from the exterior window of the inguinal canal there. And you can see the contents of the canal listed here. It's going to include the vas deferens. It's going to include what's called the gonadal artery, or in males sometimes called the testicular artery. It's going to include the testicular nerve, the cremaster muscle we talked about before, the sort of wispy cylinder of muscle that affords the ability of the testes to move closer to the abdominal cavity. You can see the network of veins in there called pampiniform plexus. Both males and females have a pampiniform plexus adjacent to the gonad. And we've already talked about the important function of these between the dargus muscle and the cremaster muscle. Both the density of tissue between the testes is changed, and the relative proximity of the gonad to the abdominal cavity is altered as well. All right, let's take a break there, and we'll